Great. Good afternoon, everybody. And just as to reiterate this and say it again, Vericel was unable to present today. I don't actually know the reason for that, but we will start a little early with our next presenting company, Bone Therapeutics. And here to present for the company is Thomas Lienard, who's the CEO. And as you may know, Bone Therapeutics is a Brussels-based orthopedic regenerative medicine company focused on manufacturing osteoblasts from mesenchymal stem cells. The company's lead autologous product, which is called Preob, is in a phase 3 study for osteonecrosis of the hip. But the company is also developing an allograft product, which is a theme we're hearing pretty often today, called Allob for the spine and also delayed union fractures. So Thomas, perhaps you can start off, and I think we're going to have a little bit more of a kind of dialogue here where you use your slides to augment any answers, but perhaps you can start off by really describing the process to turn MSCs into osteoblasts, and maybe even just tell us what osteoblasts do for those of us who would be too shy to ask.
All right. Well, osteoblasts are bone-forming cells, so typically they will act together with osteoclasts, which will break bone, so they will act in combination. Osteoblasts will form bone. What we do is we use mesenchymal stem cells from the bone marrow of either the patient for our autologous product or from a healthy donor. But what we do is we just extract the mesenchymal stem cells from the bone marrow, and then in a proprietary process, we differentiate those cells into osteoblasts and expand those cells ex vivo in a process that lasts a few weeks, and then ship it back either to the patient himself or just cryopreserve the material and deliver it whenever it's needed for the allogenic process. The difference compared to what other players are doing is that our cells are already differentiated. That means they know what they are there to do, and they, which we believe makes them more potent. They're like a trained army, and also safer because they're not going to differentiate into any other type of cell.
And Thomas, just to make sure I clarify on that, you mean they're already differentiated after your proprietary process, so as the product is implanted, that's when they're differentiated?
Exactly. So the process differentiates them and expands them, and when we reimplant them in the patient, they are already differentiated in pre-osteoblasts and osteoblasts.
Great. And as I mentioned in my introductory remarks, your lead program, Preob, is an autologous product. So maybe you can start out by talking to us about the phase 2 study and the design and the data that you reported with Preob.
Yes. So Preob is the historical product. That's where everything started about 10 years ago out of the University of Brussels. And it's the most advanced. We're currently in phase 3 in osteonecrosis of the hip, but we have phase 2B results that we communicated last year. What is it that we did? The indication is a rare indication called osteonecrosis of the hip. It's a disease affecting rather young patients. There is a necrosis of the femoral head leading to a fracture and then a collapse of the femoral head, which typically leads to a total hip replacement. Total hip replacement is something you want to avoid in young patients because those prostheses have a limited life expectancy. So what we did, we analyzed 60 hips, 30 were treated with a core decompression, which is a drilling of a hole basically in the femoral head, and Preob, our autologous product, and 30 of them were treated with core decompression and bone marrow concentrate, which is an active product. What we looked at is the proportion of responders. A responder is someone who did not fracture over the 2-year period and experienced clinically relevant reduction in pain. So if you do not fracture over 2 years and you have less pain, you're a winner, you're a good patient. What we see, as you can see on the slide, is that 70% of the pre-op patients were responders after 2 years versus only 37% with bone marrow concentrate. And remember, bone marrow concentrate is an active component. And we see the same trend at 3 years. When we looked at progression to fracture only, so the percentage of patients that fractured over the period of 2 years and 3 years, we see that 20% of the pre-op patients had fractured versus 40% of the bone marrow concentrate patients. So there's really a 1 to 2 ratio. And this gap is even wider at 3 years, where you have 20% of the pre-op patients fracturing versus 50% of the bone marrow concentrate patients. So we believe that this product is safe, safety was also good, but also efficacious, and that those cells are very much producing bone.
Very interesting. Now, I have kind of I apologize, a dumb question. So why not just get a hip transplant? Or why not just get an implant? And I think I can imagine the answers, but maybe you can also tell us, are patients today, do they typically go to fracture, or are they closely controlled by their docs? They experience pain, other things. They know they have osteonecrosis of the hip, and therefore they kind of keep an eye on it, and then they say, you know, we need to do something about this before you fracture. So maybe tell us a little bit about sort of the patient's path, and why wouldn't they just get a hip replacement?
Yeah. You want to avoid a hip replacement in those patients because they're young. Typically, they're 35 to 50-year-old. A hip has an expected lifetime of 10, 15, 20 years maybe. You can replace a hip once. It's very difficult to replace it a second time. So if you're 85-year-old, it's fine. You can get your prosthesis, you'll be fine.
And they're less active.
And you're less active. If you're 35, the story is different. So physicians want to avoid that procedure or delay that procedure. At this time, there is actually not much that physicians can do for those patients. Yes, they experience pain and they know they have osteonecrosis of the hip. What they do is core decompression. What they do is, pain relief, these kind of things. But really, changing the progression of the disease is still very difficult.
That's helpful. I appreciate you answering that question. So let's roll forward to the phase 3 study. Tell us what you're doing and when we could get data for that.
Yeah. So in the phase 3 study, very similar design, except that we are, the comparator arm is not bone marrow concentrated, but it's placebo, core decompression plus placebo. So we are expecting interim data most probably next year. So we'll have a very good idea of where we are in that phase 3. And this is really the objective with the autologous product, is to deliver phase 3 data that prove that our bone-forming cell therapy or technology definitely works. Our strategic focus is much more on the allogeneic product, because we believe that this is where the real potential of the product is, with a product that's easier to scale up, easier to produce for large patient populations.
So maybe let's make that transition now, and I appreciate you kind of giving us the pre-op story a bit as background. So where are we with the allob product now, and other than obviously being allogeneic versus autologous, how is it different than pre-op?
Yeah. So the advantage of allob, the allogeneic platform, is that, well, it's much closer to a pharmaceutical or biological product because you have an off-the-shelf product available. With one bone marrow, we produce many different doses. And the product is off the shelf. It can be used whenever it's needed. With our product allob, we're targeting 2 indications, and let me try to, yes, let me show you this. Yeah. We're targeting 2 indications, 2 main indications. First one is lumbar spinal fusion. We're currently in phase 2A with that trial. And the second one is delayed union fractures, so severe fractures that are not healing in a normal time. We're also in phase 2A with that trial. Can maybe show you some of our data on these two trials. Yes. So spinal fusion is a typical procedure, very common actually. There are 500,000 procedures in the US and Western Europe alone, where the disc gets, the intervertebral disc gets removed because it's diseased, and replaced by plastic cages and some material like bioceramic granules. The idea is to create a fusion between the two vertebrae so that the pain disappears. The problem is that in about 25% of the cases, that fusion does not happen, which leaves the physician and the patient with a failed procedure in the spine, which is very complex, obviously, and a patient that's still in pain. So what we do is we come and mix our cells, our allogenic cells, allob, with the bioceramic granules to promote the bone formation so that the fusions happen faster and better. And this is typically something that physicians are looking for. There have been products on the market that, growth factors actually called BMP2s, that have been very successful. Well, they were confronted with several safety issues later on, but the message is there is really a need for those products. So in this indication, so we're in indication that where there is a high unmet need, a very large indication, we will communicate results on 16 patients in September. It's an interim analysis of this phase 2A. Here you can see results on the first 8 patients that were very positive, both from a safety and efficacy perspective, radiological efficacy, but also clinical efficacy. And here you have images of a patient where you can see that after 6 months already, and also after 12 months, you see a very nice bony bridge being created between the two vertebrae, which means that the fusion has happened. And that's very much what you want to see.
Excellent. That's really helpful. Now, you said we're going to get the 16-patient interim analysis. When could we get full data from that phase 2A study in the spine, and what would be next steps?
Yeah, in the spine, we'll get the full data analysis probably towards the end of 2018. Obviously, we always need to wait for 12 months after the last patient has been included. So that's what we expect. And then we'll move on to next steps in the development. With our allogenic product, Allob, what we're doing is really working on making this product a really commercializable product with high yields, easy to produce, easy to use, and also cheap to produce. So we're very much working on pushing the product in a direction that makes it a real commercializable product.
Excellent.
So if we're looking at delayed union, which is the next indication we're busy with with Allob, the allogenic product, here we're looking at patients that have severe fractures, severe unhealed fractures. So typically high impact fractures like car accidents, sports accidents, so can be young patients. Typically a fracture would heal in 3 months. If it doesn't, it's called a delayed union fracture. What we do here is we just come in a minimally invasive way, so just through an injection with a needle, with a trephine, and we inject our cells in the fracture interline. And the cells, what are our cells going to do? They're going to do 2 things. First, they will replace the defective or the missing cells. That's one thing. But most importantly, they will act as a growth factor factory. Really, the paracrine effect is really the effect that we will see, where by the secretion of factors, they will restore a healthy bone environment and recruit the patient's own cell to relaunch the bone forming process. And that's very much what you want to see. It's not just external cells doing the job, but it's really relaunching the whole machine. Here we have results on 8 patients. 7 out of the first 8 patients have met the primary endpoints. We're expecting results on 16 patients with a 6 months follow-up in September as well. So September will be a heavy month in terms of clinical results. You can see here the images, an example of it's actually the first patient that was treated. It was an ice skater, got a torsion fracture, so the foot stayed in the ice and the body turned, so not good. You can see that after 3 months at baseline, well, after more than 3 months after the fracture had not healed at all. You can see that the leg is totally broken. After the injection at 3 months and certainly at 6 months, you see a very nice progression of the healing. and at 6 months, actually, it's healed.
Great. Now, let me just ask you kind of a high-level question. What are your partnering plans for these products, either in the U.S., in Europe, overseas? How have you thought about that?
So currently our trials are taking place in Europe, so we know how to run trials in Europe, and we can run trials in Europe. For commercialization, we will be looking for commercialization partners, people specialized in this orthopedics world. For Japan, we're looking at licenses, so we're discussing with various people for licenses because we will not develop ourselves in Japan. For the US, we're looking at co-development partners and commercialization partners, obviously.
Great, that's helpful. And with the time we have left, you are a publicly traded company. How much cash do you guys currently have, and how long will that last the company?
Yes, so we're indeed publicly traded on the Brussels and Paris stock exchanges. We have a runway until about mid-2018. We have a burn rate of about 15 million euros. We had about 20 million at the end of 2016, to 20 million euros left in cash, which means we will raise money this year, most probably September, October, after the clinical results that we discussed.
Great. Excellent. I'll see if there's any questions from the audience. Thomas, thank you very much for joining us. Good luck with everything in September. It's going to be exciting, and I'm wishing you all the best.